I never addressed the topic of evolution, so to see that addressed, I mean, it makes me so happy. It makes me so happy, and I hope to bring in more of that on my channel. Evolution is harder for me to address because I'm not a biochemist, and so it's even though I ask my biochemist friends, I tell them, I don't understand the chemistry. And as soon as I start probing them on the chemistry, everything begins to wither around the edges. And so I know how to make, I know how to, when people are speaking about evolution, I know how to make them squirm just by asking questions. So I'm glad to see all of these arguments coming forward. And I really enjoyed this talk by Rob about these different standards of credibility of data, and it shows you that everything that they have is by an ultra low standard, and they seem to try to explain everything in the way of evolution. So I enjoyed that very much. I hope to bring in more of that type of argument onto my channel and expand it in that way through the experts.
Dr. Tour, I,
Yes.
I'm I'm Salvador Cordova. I was Dr. Sanford's research assistant, one of them. I just wanted to introduce myself.
Oh, nice to meet you. Yes, I've heard your name before, and I really admire Dr. Sanford. I think he's done great work. I've presented his work on it on several times to many groups. I find it fascinating.
I would be happy to share what we've done. We've actually broken the peer-reviewed barrier to criticize evolutionary biology. It was, in my opinion, a miracle of God.
Well,
And
yeah. If you would send me an email and we'll get together and we'll try to pull something together for that. That'd be great.
I am researching I'm in a PhD program in biomolecular engineering,
Okay.
and I'm actually very interested in quasi-particles. So I talked this afternoon on quasi-particles, and I know graphene has some interesting quasi-particles that I'm also interested in. So we
I
could have a
see.
very interesting conversation.
I see.
Thank you.
Thank you.
Uh, I'm interested a bit. Don't know if we covered much of it, but there's probably also some cosmological arguments. And I know that might not be anybody's area of expertise, but does anybody have maybe input on that to kind of see if we can round that off, round that off on all the topics that we've covered so far? Like Setterfield, I think, has some
Get the
some
D
ideas.
Russell, get D Russell Humphreys to talk about it, and a few of these physicists that experts in that area. We could get them as part of a seminar. It's a little bit deeper than what I want to plunge into, but I wanted to thank Dr. Tour because my original dive into creationism came about as a result of trying to make the peptide bond work in with amino acids. I figured, well, if you have a prebiotic soup with amino acids and you put them together with a little bit of electrical spark, like you had in the Miller-Urey experiment, that I would end up with getting some polypeptides to form. Well, it didn't work with glycine and alanine, but it turned tryptophan and tyrosine brown. And so what I did was actually just destroy the side chains. So
Yes, yes. Yeah, that's what you do. People don't appreciate this, Douglas, until they actually do chemistry. I hear people talk chemistry all the time, and I'm like, you've never really made anything, have you? And the problem with biologists is they just buy kits and they think it's easy. Those kits were made by chemists, and it says, mix part A and part B, and they got it. They don't realize what molecules in their native state do when it's tough. Organic synthesis is tough, and Yeah, so the synthetic chemists really see what I see. They see it. Can you imagine living the lives that these guys have to go through, living this lie, because they they see these problems firsthand. Because you tried the reaction, you did it.
Well, I had to have a encounter with God to show me a little bit of humility to admit that this is something that wasn't at all possible. Even the simplest of the chemistry to create polypeptides was not possible at all. And
Yeah,
then
yeah,
stereoisomer problem Rear just had up, and is Dr. James Coppage and his work early on that convinced me that the possibility of even having one left-handed amino acid purified in this form with a sufficient yield wasn't going to give us anything that's going to produce life.
hmm
I wasn't popular on campus after I discovered this. I
yeah,
happened to meet a fellow by the name of Dr. John N. Moore, who ended up being one of the managing editors of the Creation Research Society, and
I see.
he sort of helped straighten me out. This was back in
Yeah,
1967 that I started digging into this.
I see. Now, Crystal, did you have a question?
Yeah, I've got a question here from Walt. He wants to know, why do Christians use methodological naturalism to explain the supernatural origin of the universe and Earth?
Well, I can tell you why I do this, because I'm trying to reach an audience that's not Christian. I can go to a Christian audience and say, in 6 days, God created the heavens and the earth and all that is in it. I can say that. I can't go in my classroom and say that. And so my target audience is very different. And if you see the way Paul addressed different audiences, he certainly did. When he addressed Jews, he addressed them very differently than when he addressed Gentiles. When he went to the Stoics, the skeptics, what did he do? He said, I was walking around and I was looking at your objects of worship, and I saw that one that had that plateau there, and there was nothing on it, and it said, to an unknown God. And let me tell you about this unknown God now. Can you imagine if any pastor took an idol and used it as a jump off point to describe our God? We would have said that was sacrilegious. You never should have done it. Well, Paul did that. Paul took this empty location and said, that to your unknown God, let me tell you about that unknown God. It was an amazing transition. So we need to look at who our audience is, and we come with transitions in a way. And I'll tell you that if I go 1 week without leading somebody to the Lord in a one-on-one conversation, somebody who went from total unbeliever, didn't believe in the resurrection of Jesus Christ, to a believer, if I go 1 week, I feel it's been a wasted week for me. And I get everyone that I lead to the Lord, I set them up for a 13-week Bible study and daily reading in the scriptures. And in 2024, there were 86 people that I led to the Lord. So nobody can tell me that this methodology doesn't work. It's bringing in people over and over again. The year before that, 2023, was 107 people. And I get messages all the time that I've come back to the Lord because of listening to your content. So I think there's good reason to address it in this way and use this as a launch point for the gospel.
Well, 1 Corinthians 9 says, I become all things to all men, so that I might save some. Now, I happen to be one of those skeptical people who did want evidence and did want science, and I didn't think it was out there. So an apologetic approach, a scientific approach, it was helpful to my conversion because I needed that. Now, other people might be emotional, I would think, and maybe they could watch this and watch this whole summit and look at all the wonderful evidence that was presented, and they're still not swayed, but it's because they have there's like a heart issue, and then that might be a different approach to reach them.
I wanted to add, in mathematics, a lot of our proofs are starting off by assuming something we're trying to disprove. We call that proof by contradiction. So when I teach this stuff, I'll say, assume this is true. So I'm not saying that I believe it. I'm actually assuming it just for the sake of argument to show that it will lead to contradictions. It'll either show a contradiction with itself, or it will contradict experimental observations if you keep assuming it. And so that's why I assume I'll tell people when I'm teaching this that I'll assume this is a natural progression, or I'll assume common descent for the sake of argument. And so because I've seen it in math, a lot of the most powerful proofs, you actually start by assuming something just for the sake of argument, and you're able to demonstrate its falsehood.
You know, I was thinking about methodological naturalism and what Dr. Tour mentioned about facts being no problem with the Bible with regard to facts, but theories are different and they change. So I was thinking about the rejection of the supernatural. It seems that if we go back in history a few centuries, then I think of what the science fiction author said, that any sufficiently advanced technology appears as magic. So if we go back a few centuries and we deal with some of the scientists back then, some things like, well, just simply electrical force, magnetic force action through a distance. You know, if they were not familiar with lodestone, they might reject that and just push it out. Why? Because I'm thinking that it's not part of their scientific body of knowledge, of understanding. So they reject anything beyond that, which is, to me, seems pretty unscientific. It reminds me of what I've heard about babies. If, you know, when you play peekaboo with a baby, you hide your face, then you show your face, then you hide it. If the baby cannot see your face, the baby thinks it does not exist. So to the child, I've heard this, whether, you know, uh, that, you know, to the child, if if you don't see it, it doesn't exist. And I'm thinking about science as in the broad sense, as an institution. You know, ages ago, I've read about things like it's impossible for a human to travel faster than 20 miles per hour in a vehicle, if we go back, or to fly. And I think that's actually, it seems what we're dealing with in the rejection of the supernatural, it's the same type of phenomenon of rejecting anything not yet completely understood. I'd like to hear any other thoughts about that. Tell me if I'm wrong too. I could be wrong, but that's what it looks like to me is happening. Well, anyway, I guess I will say a little bit more too. Something else Dr. Tour mentioned about theories. It seems that the theories being changing. It seems to me that one of the problems in science, in the history of science, might be that a theory, maybe a good theory, maybe a theory that seems to fit the data, the evidence, at least the limited set of the real data that happened to be known and understood at that point in time. Okay, so instead of saying it's a good theory, the scientific world says it's reality. And then when new evidence comes in later, it makes it more difficult to dislodge that theory, because now it's not rejecting a theory, it's rejecting of reality. And that makes it maybe psychologically a bit harder to do. So I think it's really helpful to keep theories as theories, like the theory of evolution.
I think that's a definitely a good point, because evolution's presented as fact, right? And so I was somebody who used to believe in evolution. Is because all of the education, all of the formal education I was receiving, either from public school or even attending college, only taught evolution. I never heard another perspective. I didn't even really know there was another perspective, other than they demonized, oh, there's those weird Christians who think the earth is only a few thousand years old. That's all I ever heard about it. So the opposing view was demonized, and the one side was presented as fact, regardless of the institution. So how can anybody really think different? And why would you even question your educators, who you already look up to as an authority? But we do have, I think, a couple of questions out there.
Yes, we do. We
Oh,
have
there.
one here. Just bear with me a second. Screen is moving quicker than it was before. So we have a question from Donnie with Standing for Truth. He asks, is nylon digestion by bacteria a good example, as asserted by evolutionary scientists, of an information adding complexity increasing mutation?
I can take that one because specifically Dr. Sanford and I wrote a paper on that. We're still trying to get it through peer review after 7 years. And I actually have a chemical diagram here if I could share it. Since we have organic and biochemists here, I would be honored to have them do a little peer review right here for you, because I anticipated it, because I actually saw it on the answer is it's not a good, we have refuted it. One thing was because I had studied at the NIH, because Dr. Sanford wanted me to study there. And one of Dr. Kunin's staff workers was my professor, and I asked him, and he looked at this. He said, "Yeah, definitely, you're right. Ono was wrong. That has been a myth that should have been, that should be falsified by now." And we've sent this in to peer reviewers. They said, "Well, we're not going to do this because it's obvious that this is false, so we're not going to publish it." So they agree with us and say, "Well, it's nothing new." I don't know. Can you see this? It says testing the hypothesis, the nyelinase NyLB protein.
Mm-hmm.
Do you see that?
Yes.
All the chemists in the group should recognize this. This is lysine. And this reminds me of something that was in the debate between Dave Farina and James Tour, and how you get a amide bond where it shouldn't happen. This is a generic peptide bond. So the lysine kind of connects in a way it shouldn't. And so it's very easy for bacteria to digest this. All you need is an amidase or peptidase. We have since found out that humans have trypsin, which can break the nylon bonds. And so pigs also have trypsin. So that's really nothing new. It's just a matter of whether you could digest the residues, the individual residues. We're going to try to pull on a biochemist to be a third author and try to get this through peer review. We can't, because we've been sending it to evolutionary biologists. They keep shooting us down. The editor will say, "Well, hey, you're right, but this isn't news. Some people have already figured this out. We're not interested," or they'll come up with some other excuse. We have an editor interested now on this topic. This is not a difficult reaction. Trypsin, even papaya can break this down. We went through a lot of bioinformatic details. This is a huge paper. It took us a year to put together. One thing we know is that this capability exists. It's very far wide. And what they say in evolution, they'll say that this is ancient. It had pre-existed the existence of nylon. So I hope that helps answer the question. If you look up that title, you'll see it in the chemical archive, and you can read up on it. I've mentioned it several times. I've had the editor of Cell Reports look at this. He said, "Well, we don't like the style of your paper, but your conclusion's right." So I hope that's a long-winded answer.
I have a question, and that has to do with whether or not undergraduate students in the university really get a chance to experience science at all. We have, I was a chemistry major, and I remember being frustrated because, we were in class in the laboratory doing cookbook chemistry, and we were just doing the experiments that were out of the book. But to be able to actually explore a problem and to actually do a scientific test on a hypothesis and to be able to actually test something. This was something that I had the privilege of having a chemistry professor that I had in high school, where they said, you devise your own experiment and try this out and see if it works. And that's where I did my little experiment on amino acids. But I didn't see in universities do anything except the indoctrination and cookbook chemistry. I see this as a problem with our education system, that we're not able to really experience what science is all about.
Well, I can speak for Rice University that we require our science students to get into a research lab. And as faculty, we put them right alongside our graduate students and postdocs, so they certainly have that opportunity. Some do more with it than others, but I have many undergraduates that are co-publishers on papers. So I think that they certainly do get the opportunity.
That would be true at Colorado State University as well, Department of Biochemistry. So I think it depends on the university. Typically, smaller universities don't have as much research as some of the larger ones, so maybe the opportunity is less. But even at CSU, I run a NSF-funded RAU program. It's called Research Experience for Undergraduates, where we bring students from smaller universities to our university and give them the opportunity over the summer to participate in vigorous research and research labs. So yeah, I think that the opportunities are there. Students have to have the initiative to find them and take them, but they're certainly there. Faculty in our department is very open to that.
Now I have a question, especially Dr. Sholders and Dr. Tour. Since you are currently working at universities, are these secular universities? And does that pose any sort of, is there anything you run into having the viewpoints and the worldview that you have? And do you get any pushback or discrimination, anything like that?
Well, I talk a lot about Jesus. That bothers people probably more than talking about origin of life. But what I've come to see is that if you talk a little bit about Jesus, there'll be pushback. But if you talk a lot, they leave you alone because they don't want to get you started. And then origin of life. A couple years ago, I was invited by a student group, Ratio Christi, to give a talk on origin of life. And for some reason, there was a new person on the Rice, in the Rice Public Affairs office, and they saw, oh, this professor is going to give this evening lecture. So she put it up on the Rice Instagram feed. And there were people all over the country, Rice, alumni and students. Oh, this guy's going to talk about flat Earth, everything. So when I got there, they said they apologized to me for all this. I said, I didn't see anything. I don't read Instagram. And so I gave this talk, and it was very well attended. The place was packed, and it went very well. And then the school newspaper was going to write an article about how I was talking about silly things or something. And then I put it up on YouTube, and it exploded. I mean, tens of thousands of views night after night after night, and really took off. And I told the newspaper, if you want to write an article like that, I'm going to do a YouTube article on the editors of the school magazine and the way you guys write about us. And they said, oh, please don't do that. And so they didn't write it. But and I think this has gotten 300 or 400,000 views now. And so these things can really take off. And nobody's come and said your science was wrong. It was a derivative of the talk I gave tonight. Nobody has said your science is wrong. So sometimes there's pushback, but Jesus did say, blessed are you when men revile you and persecute you and say all manner of evil against you falsely on account of me. Rejoice and be glad, for your reward in heaven is great. For so they persecuted the prophets who were before you. Woe to you if all men speak well of you, for so they spoke of the false prophets. So Jesus warned us this would happen. And I think that we have to be careful not to flee from these things. In John chapter 12, it says, many even of the Pharisees believed on him. but many even of the religious leaders believed on him, but they were not confessing him for fear they'd be thrown out of the synagogue. Now, thrown out of the synagogue is a big deal. It's not like you go across the street to the next church. I mean, you have no contact with anybody anymore. So you'd think that the Bible kind of understands the pressures. But interestingly, the next verse says, because they love the approval of men more than the approval of God. So this is pretty serious stuff. Do we love the approval of men more than the approval of God? As believers, are we going to take a stand on things? Are we going to take a stand for Jesus? And as you guys know, I've taken quite a lot of flack for taking my positions on origin of life and challenging the dogmas on this thing. Sometimes it has robbed my sleep from me, but I got to suck it up and move on. And Jesus bore a lot more, and Paul bore a lot more, and people are getting saved. So this is what we do. And I think if we sugarcoat it and say, well, God understands because this might happen in your career. Well, did God understand when John wrote, because they love the approval of men more than the approval of God? I mean, so it doesn't seem like there's a whole lot of patience for not speaking up.
Yeah, I mean, at Colorado State University, I've been told that I have a file on me of complaints for my views. I was called to meet with the vice president of diversity, equity, and inclusion, and because I take the opportunity to at the beginning of my courses to introduce myself as a Christian and describe that means that I believe that Jesus Christ was the creator of the world and died on the cross and rose from the dead and is coming back to set things right. I kind of give the gospel, and a lot of students don't like that, and so they complain, and then I guess there's a file of complaints on me. So nothing's risen to the level of what I would call persecution in regards to you're going to lose your job if you continue to do this, or you can't do that. I've never actually been told that I can't do that. So, but I've had quite a bit of pushback on it, as I think would be expected at a university that's secular. At the same time, I've had lots of students come up to me and say, never had a professor do that. And so encouraging that you would do that. Thank you for doing that. I've had lots of students that are not believers that have come and engaged and talked to me about those beliefs. So it is what we do, and we expect it. I think the temptation, for all of us, but myself in particular, is to find the approval of man instead of God. And so that's something that we have to actively come against. And certainly for myself, that's something I have to actively come against. So
thank you both for that. Now looks like we have a few more questions in the comments. Is that right, Crystal?
We do. So our next one is from Wesley Coleman. He said, is there a problem for evolutionists in that the stochastic processes in DNA are regulated and their advantages are understood to fit an environment? He said he knows it's a loaded question. But he'd like to hear your response.
Could you read the last sentence again? I didn't quite catch the last sentence.
Sure, I'll read the whole question again. Is there a problem for evolutionists in that the stochastic processes in DNA are regulated and their advantages are understood to fit an environment?
I would say I don't know. Maybe I don't fully understand the question. But yeah, I don't know the answer to that.
Could that be referring to punctuated equilibrium? Because I'm thinking of punctuated equilibrium, Stephen Jay Gould's theory with Niles Eldredge, but that seems to deal with, species, not down at the level of the DNA, although theoretically they should be, they seem to be related in evolution theory. Could that be what the question's about?
I don't worry about the materialists, evolutionists' problems if they have problems with things. I mean, they have to overlook so many things. It's not just that. I don't worry a whole lot about what their problems are. They're having difficulty with so many things. It's probably one of many, maybe, but they just continue to put their head down and plod along. They've got an echo chamber, so they just continue on.
Fred, were you hoping to chime in there before I went on to the next question, or you good?
One of the things I was going to just say really quick, I don't know if they're referring to the stochastic process that God designed into the immune system to target things the immune system is attacking, because it's pretty, I think some of the other speakers could better speak to it, but maybe that's what they're asking about. Because I know there is a design behind using the stochastic process to identify invaders, and then remember, because it makes it for a faster way, I believe, to find invading antibodies or invading pathogens.
Yeah, so I put it out there if you wanted to elaborate on his question that he can. For now, I'll go back to another one that he has. If proteins do not have common ancestors, do protein orchards prelude common ancestry?
I'll take that one because I'm the one who brought it up. When I make this argument, I say if you assume common ancestry, you're going to have these difficulties. So on the left is a collagen, and I highlighted the glycine residues in there. And then on the right is a zinc finger 136. I highlighted the cysteine and histidine residues. It's a striking pattern. And even a Rutgers evolutionary biologist by the name of Dr. Dan Stern Cardinal, I said, I'll just grant you, the presupposition that there is a universal common ancestor for cells. And I played this clip before. He said, for those who missed it, can I play this clip briefly? It's only like 30 seconds. Let me reshare it with the audio. It's only 30 seconds, but this is an evolutionary biologist. And then, just for the audience that's not aware of Wesley's question, this is it.
On that, we agree. As you said, like we agree that proteins, unlike cellular life, do not share universal common ancestry, right? We disagree about cellular life, but we agree that proteins don't share universal common ancestry.
Proteins don't share universal common ancestry.
Okay, so that was a little, okay, Fred's laughing. That's a little, okay, so I said, hey, look, I'll meet you halfway. Let's just assume for the sake of argument, that there is cellular universal common ancestry. But I said that the problem is you're going to have to deal with, let me get my screen share again. You're going to have to deal with the problem of these proteins. They're very complex that would just have to come out of nowhere. So it's not like it's a great victory for them, because it actually has some bearing on the origin of life. Because there was a show on the amyloid hypothesis recently with Dr. Truman and Dr. Tour. In a debate, I confronted Grayson because I debated him. And I said, how's your amyloid going to evolve into any of the other proteins like topoisomerases, helicases, polymerases? They're all distinct. So it's like you're going to have to invoke multiple independent routes to that. You could assume common ancestry, but it's going to cause you a lot of problems, especially at the early stages of the formation of the cell. Now, if you assume it later on, so for things like collagen, which I'm going to try to get this back up. The collagen, you're not going to have a lot of animal metazoans. This would just have to pop out of nowhere. And so I try not to push the argument too far and say, well, this absolutely refutes universal common ancestry, but let's just assume it. You're going to have to invoke miracles, statistical miracles along the way to make all of these. So, at first when I presented this to Dr. Dan, he said, I think you're right. But after 4 years, he said, yeah, you're definitely right. You could see the spelling of it is very distinct. The shapes are very distinct, just like parts of a car. They're all very distinct. So I would just, I would not try to push this to refute universal common ancestor. I think this is damaging enough to try to be doing this. So, so people have tried, because they like that argument, to use that as evidence against universal common ancestry. I'll just say, no, I think it's a victory enough to say the proteins don't share a universal common ancestor. This is going to be very problematic for them. I don't think they realize it. A lot of evolutionary biologists are not protein biologists. They don't know the difficulty of making some of these things. The amount of technology it takes to make a topoisomerase or a helicase or polymerase. It boggles the mind. And now that we have AlphaFold is trained on these data sets on how a protein will fold. We can't actually build these things, from first principles. It's just too difficult. We don't have the technology. We can't possibly do it. So in the ID community, we're circulating the idea that God made all of these proteins as oracles to teach us how to do chemistry and nanotechnology, because some of the stuff is just unsolvable by regular means. So I wouldn't push it to try to refute universal common ancestry. There are probably other ways to do it. If the earth is young, it's done. So that's what I say.
Awesome. Thank you so much for that. Did anybody else want to chime in, or are we good? He's given us a little bit more emphasis on his last question there. So he said Fred got part of it. He's referring to the random mutations generally, which are purported to be a, quote, creative mechanism.
Hey, I'd like to jump in on this one. I'm sorry, I'm not sharing video right now. So I'm not sure if this is what he's referring to, but this is what it reminded me of. And I think it's a really, really important way to reframe how we look at mutations in terms of adaptation and biological evolution in its simplest sense of change over time. And I would like to give credit for folks like Dr. Galuza, Randy Galuza, for helping me reframe how I think about mutation and random mutation. I was, as being educated in secular universities, sometimes I borrow their language and I talk about adaptation in a biblical sense as being a very random process, and you get random mutations which are generally deleterious, but that can still be a benefit to the organism, and then those mutations are naturally selected, and so you get this some people call it microevolution, adaptation, etc. And so there's still this place for these random mutations in a biblical worldview. And what I've been encouraged to think and consider them more as now as that the process that we probably shouldn't even call them mutation. Certainly many are, but there's probably a designed process for change in the genome that is an engineered process, just like we see engineering and all the other things that were talked about today. There's probably an engineered process for adaptation and creating non-random mutations, which we might be calling random mutations, and they might not be as random as we think. And so the Institute for Creation Research calls this continuous environmental tracking. Instead of this blind random process of mutation and adaptation, is actually a designed process of organisms responding to their environment and adapting. Certainly, that is a very different way to look at this process of mutation as not so much of a random thing. Now, don't get me wrong, there certainly is a random component to mutations, and there always will be. But we should probably look at it also in light of a designed change process.
Dr. Brown, was there some research about that regarding involving fish, blind fish?
So that's exactly what Randy Galuza and the team there at ICR is working on. So this is a perfect example. So you have all of these. You have cavefish, which are they have lost their eyesight. They've lost their pigmentation because they live in caves underwater that there's no need for those things. But then there are seeing and pigmented populations of those same species that are above. They're in water that is exposed to light, and so they do have eyes and they do have pigmentation. And so this same pattern has occurred, the loss of the eyes, the loss of the pigmentation, not just in one population of cavefish, but independently in many populations of cavefish. So evolutionists look at this, they call that convergent evolution, different populations converging on the same thing. Some creationists have looked at this and I think rightly said, wait, this looks like there could be something designed here. In other words, these organisms are seeing are seeing, they're sensing a change in their environment, and they are reproducibly adapting in accordance to that. So it's a designed adaptation. And let's be fair, we've got a long ways. Kudos to ICR for taking this on. It's a bold hypothesis, but even if it's partly true, that's a really intriguing way to look at adaptation.
Yeah, Wesley had said that you gave a nail on the head answer for his question as well.
You know, I was thinking on the abiogenesis thing. So even if somebody were to answer to your challenge and were somehow able to create life in a lab, all they proved was that it took intelligence to create life. Because to really prove it, you'd have to pour some chemicals in a room and let it be and see if life comes. You know what I mean? Like it couldn't be guided.
Sure. I mean, making it in a lab today, even under prebiotically relevant terms, doesn't say that that's how it formed the first time. Yeah. And so, you know, you'd have to say at a hydrothermal vent or in a tidal pool or in a rock somewhere. And makes it very hard. But, you know, Kurt, you and I don't have to worry about this. It's not going to happen in our lifetime. It's not going to happen in our children's lifetime. It's a long way away.
Yeah, Jesus will come back before somebody answers your challenge, I think.
Probably.
I've got a question that might be quite interesting to discuss. I was, and this might be something Dr. Brown can answer because he lives in Arizona. I visited Montezuma Well, which is near between Phoenix and Flagstaff, and it has like a carbon dioxide environment. It's got a sort of an interesting chemistry in this little lake. And in that lake, there are 5 endemic species that sort of feed upon each other. There's a diatom, an amphipod, a spring snail, and a leech that all have a life cycle where they feed upon each other. They exist nowhere else in the world. Now, how is this possible? How do we explain this from a creationist point of view?
Hey, so since you referenced me in that question, I'll jump in here. So first of all, I'm not familiar with this particular well that you're speaking of. I've just recent transplant to the Phoenix area, so I'll have to explore that myself. But I think in general, this is a question that comes up. This is a very, very specific example of this, but the same thing about freshwater fish adaptation, post-flood, saltwater fish adaptations, post-flood. I think what we see in general, and I'm talking in big terms here, is that in a post-flood environment, you have things that there's a selection that occurred just in the survivors, so only a subpopulation survives, and then there's kind of a fine tuning to a particular environment. So this dependence that we see now likely did not exist initially when that ecosystem came about post flood. That's how I would look at that.
We'll have to talk about that when I come out and visit you coming up in a few weeks.
Absolutely. Maybe we can go on a field trip there.
All right, I think Sal has a question for everybody, all the chemists in the group.
I've been trying to find chemists, and I don't know if Dr. Brown can help. I'm trying to find chemists or laboratories that can measure the level of homochirality. I know Jeffrey Bada, National Academy, had run the experiments, but they'd been digging oil cores, and they're finding, well, in addition to, they find peptide bonds in these old biotic materials. As far as I've been tracing, we're seeing homochiral forms all the way down to the maybe Cambrian, and no one has bothered researching because they've assumed it can't exist. I mean, this is one reason, because I study Gibbs free energy and kind of primitive origin of life stuff. that's kind of an easy reaction because there's stuff doesn't stay homochiral for very long. So people gave up. They said it must be contamination when you find fossils with homochiral proteins. And I've been looking for labs. I've been asking around. I mean, is the technique very expensive to test for that? Are there laboratories that can do this? Does anyone know? Have contacts?
Are you looking for it in amino acids, or are you looking for it when they're already in polymers? In other words, peptide bonded structures.
Either. The ones that are unbound have very similar profiles to the ones that are bound. I think the bound ones last a little longer. The free amino acids flip their chirality easier, but we found some that are still free floating. So whichever's easier, I'd be happy to start.
Oh, it's very easy to tell if it's just a free amino acid. I mean, there's chiral HPLC. Boom! You know that immediately. Yeah.
Is it an expensive test? I mean,
expensive is a relative thing. Who's paying? So I don't know how much it costs. There's a lot of commercial labs that can do that. My guess is $50 a sample, something like that, if it's a pure sample. But if you've got a lot of other things in it, it makes it harder because it can confound these things, and they don't like to inject garbage onto their columns.
okay.
So it's just going to depend what's there. Once it's in the polymer chain, it makes it harder to figure it out. You would probably use a degradation mechanism and then you use some technique that does not induce racemization during the degradation mechanism, and then look at it as the individuals come off.
So the degradation would be breaking up the peptide bond. So we could probably take a sample of a known protein from today, break it up and see.
Yeah.
Okay, all right.
Yeah, and if you can degrade it, I mean, the degradation is going to be more difficult if you've scrambled a bond, because the enzymes that do the degradation are not going to be able to recognize when the bond has been racemized. But again, I'm speaking, I don't work in that area, but just dealing as a synthetic chemist. I mean, this is not a hard problem to do. If you couldn't find anybody, you're talking to the wrong people. But that's not something that I do, but it's a pretty standard type of chemistry.
Sal, can you clarify what samples then would you be interested in testing?
We found, okay, so Jeffrey Botta, this is published Proceedings, National Academy of Sciences. They're actually very troubled about the lack of racemization in fossils. They said this had to be contamination, and they were taking seashell samples. And the reason I laugh is it's kind of the same problem arising when we're finding peptide bonds in dinosaurs. And just like, well, we could do all sorts of things with racemization, and we could also, we don't have to be restricted to dinosaur bones. I'm sorry, Dr. Brown, what was your question again? I totally lost track of it.
No, I was just following up with the samples that you are interested in measuring. Like what's your source of those proteins?
I would like some of these so-called living fossils that are actually fossilized, like say down in the Cambrian. Because I found an old 1960s paper that was saying, hey, we found some homochiral amino acids in the Cambrian, and no one's followed up on it for its obvious implications. Their dinosaur bones are hard to find, but there's so many shells, and there are proteins inside those shells. And I've been trying to advocate to just dig these up. We could do preliminary tests. I mean, everyone's so excited about dinosaurs, but there are lots of living fossil bones. We could do this. This is also interesting chemistry. So maybe I could talk offline with you, Dr. Brown, plus some other projects.
Yeah, no, I think we should follow up with that because, it's certainly in line with the sorts of things that we do. We don't do those techniques, but we work ourselves and we work with other people who are dealing with ancient biomolecules, DNA or protein or otherwise. So, yeah, it's along those lines. We should definitely follow up.
Awesome. We have another question here from Wesley Coleman. He says, do you all have any good examples of clearly pro-creationist articles published in a secular peer-reviewed journal?
So I think it'd be curious to hear what Wesley means by pro-creationist article. Um, if hinting a procreation view is enough to get stonewalled, I think everyone here would probably has experienced that to some extent. So, but that being said, there's tons of data that's out there that is procreation that's published by evolutionists. For instance, the Mary Schweitzer stuff. Now, obviously, that's not they accept a conventional timeline on everything, but I look at that research and I think this is empirical research that really nicely is procreation. They just don't call it that. I think if you're talking about overtly procreation, I could not give you a single example.
I'm just wondering, Joe, why is Mary Schweitzer's work clearly pro-creation? You mean pro-young Earth, or you mean procreation?
So I'm interpreting Wesley's question as being pro articles that support a young Earth perspective or a biblical
Okay.
creation
All right,
perspective.
all right. Yeah, because there's so much beauty in biology. I would say every article on a biological system is pro-creation. It's just good. If you make molecules for a living, you look at biology and you're awestruck. It is procreation. So I think that was the question, but I just didn't understand it. But now I understand why you answered the way you did for Mary Schweitzer's papers, because you were looking at it as a pro-young Earth question.
Yeah, no, that's correct. And I often say all science is creation science, and by that I mean empirical science. I think that's what you were saying there. By anyone who studies a biological system is studying really the beauty of creation, whether
Yeah.
they recognize it as such or not.
Yeah. I mean, I look at I saw my children growing up, and I just constantly amazed at God's act of creation. So yes, every article is procreation when it comes to a biological system, and when it comes to a non-biological system, as you look at the physical constants that must have been in play for this phenomenon to happen, that's procreation. So it's like what Douglas was saying, that your theology affects your geology. I think this is something analogous to that.
Awesome. We have a few more questions here from the audience. We have someone asking, what are some good churches that teach creation near Phoenix?
They should email creationresearch.org. I can be glad to share some of that offline. I attend First Baptist Bible Church in the Phoenix area.
Awesome. Thanks for that, Joe. All right, there's another one here. This one's from Walt. Has Dr. Tor heard of the origin of life experiments by Robert Hazen? If so, what does he think of the work of Hazen?
I'm sorry, I'm not familiar with the work. It's hard for me to comment directly. What did he do that was so extraordinary?
He was a geochemist at my undergrad alma mater, George Mason. He teamed up with Harold Morowitz. These are like very old origin of life researchers. I mean, from like way back, like 20, 30 years ago, he had fought against the Urey-Miller experiment, and he wrote the book Genesis. I have an autographed copy of that. So all that I could give you of what I studied, he would be just like all the other origin life researchers saying, we're getting close. So his colleague said, about 20 years ago, we're going to solve the origin of life problem in 20 years. And I said, I'll buy you a beer if you do that. I'm not going to have to do that. I will tell you an anecdote. We organized in 2006 an idea club meeting, and Jonathan Wells of the Discovery Institute was giving a talk. Hazen came up during the Q&A, and he was turning red. he was so mad. And he was just saying, you're throwing your religion at me and all that. And he was holding this book by Barbara Forrest, Creationism's Trojan Horse. And Jonathan Wells just very calmly smiled. He said, okay, well, what if you take a cell and, and it's put in, solution with the right pH and, the nutrients or whatever, and you poke a hole in it and all its guts spill, all its insides spill out. Do you think it's ever going to reassemble again? Hazen just stormed out of the room. That's about all I know of Robert Hazen. I read his book. It seemed just like all the other origin of life stuff. But his fame as being a geochemist, because he comes up with a lot of the ideas of what the early Earth's prebiotic chemistry is. The one interesting thing is he was saying, yeah, we have these meteors that seem to have life in them. It's 4 billion years old. The bacteria inside, that's the only thing that really stood out. That's the first time I learned about that. So
yeah, I wouldn't be surprised if a bacterium could survive on a meteorite. I mean, buried inside a meteorite, I mean, bacteria are really tough, and they might survive. So it might have come here, but that's not the questions that we're trying to answer. It's the origin of first life. So to the question, then it's you answered it, Salvador, that it's more of the same. I get sent articles all the time. People send this to me. So what now? Have they got it now? Have they got it now? And there's no rush to check it.
And they're always making predictions. Oh, we're really close. We'll fix this in a few years. And the same old story.
Unless they've solved that dipeptide and disaccharide, they haven't solved it.
I find that there is a need for creationists to be multidisciplinarians, to be able to discuss in a sort of a general way, geology, physics, biology, chemistry, and have a little bit of knowledge in all of them, and as well as being familiar with the Bible. It's kind of a difficult thing for many people to do, to delve into all of them, because the biologists will appeal to the geologists will appeal to the paleontologist will appeal to the cosmologist, and then the cosmologist would appeal to the other disciplines, and they sort of dance around in this manner to avoid dealing with the fact that they need to deal with God himself and to repent.
Yeah, it's a lot to study. Like I probably fall more on the philosophical, logical, theological side in what I study, and so in that sense, we kind of all need to rely on each other in having these different levels of expertise. I think there was another question, Crystal.
Yes, and hopefully I can pronounce these words. Okay. What are everyone's opinions on Dr. Behe's concept of irreducible complexity? Have experiments with VAT pace, I think that's the word, proton pumps, mutating a 3-component versus 2-component rings refuted the theses.
I have thoughts on the irreducible complexity argument, because I do want to teach an intelligent design course. And I've seen Dr. Behe debate Dr. Dan. You saw me with Dr. Dan, who's an evolutionary biologist. Even let's just hypothetically say you found some irreducibly complex system, and it's hard to define that, by the way. You can evolve it in real time from one thing to another. Does that falsify all the other events? All the other instances. So this is like going to a casino and say you beat the dice game, the craps game once. Does that mean over time you're gonna win this? Absolutely not. And so what I've tried to tell ID proponents who are using this argument, don't say it's an irrefutable problem for biology. look at specific examples, you're going to win the argument. So my favorite's the topoisomerase, which cuts the DNA, untangles it, and then religates it. I said we can't build a nano machine as efficient as that. And if any of that's missing, it's not going to work. And I'll say, look, I don't care about any other system, and you're trying to appeal to that. You got to solve this system, otherwise life is dead. So I would use irreducible complexity. What I prefer to use is all or nothing complexity as a heuristic to find examples that are hard. I wouldn't use it as a blanket argument, because Dr. Dan was very clever. He would find something about viriporins with HIV that no one hardly knows about except him and his colleagues. And then they'll say, look, I solved this irreducibly complex system. See, your whole theory is wrong. And I'll say, well, I don't care about that. Whatever you call it, I'm trying to get you to solve this topoisomerase problem, which, by the way, no one has. But what I would suggest is just look at the Discovery Institute website. Dr. Deweese, who spent his life studying topoisomerase, he gave a 2-hour talk on this. And I've noticed no evolutionist will assail that. They went after the flagellum. They never went after the topoisomerase. So I would just kind of if I'm teaching ID, I'd say, look, this is a good historical way to understand the intelligent design argument. I wouldn't use it specifically. I would use it to help me find examples like topoisomerase. There are probably others. I like the idea of nuclear localization signals for eukaryotes, or the translocation systems. There's so many other proteins we could find, but especially the ones that are critical to cell replication. Just look at those. Even I don't know about VATP. What is it? VATP synthase, and the changing of the rings. Go with something that's tested. My favorite's the topoisomerase, and the Discovery Institute invested a lot of money to put that forward, that example. I think the reason it hasn't gotten a lot of press is the opponents don't even want to argue over it, because it's such a strong argument. There was some weakness with the flagellum. They're not going to have that weakness with topoisomerase. So that's what I'd recommend.
Awesome. Thank you for that, Joe. We just have 3 more questions if you guys have time to stick around to answer them, and then we can clue up. Might be 4. This one's from Matthew Wellborn. Dr. Tor talked about a challenge to, quote, assemble a cell in a lab. Even if this could be done, how would the cell be brought to life? And what is the spark that starts the process of life from non-life?
Nobody knows. Nobody knows. If you have everything in place, does it start going? Well, if that were the case, how come a dead cell doesn't just spontaneously come back alive? Nobody knows. This is a great question. You have everything in place. Does it just start operating? Well, then a cell just dies. What is it we just lost? It's very hard to know, but that's right. Even if you put everything in place, we have no idea whether it's going to start operating. In an automobile, you have a starter that turns the engine over, and so that you starts hitting a spark and going. I mean, you could have an entire car, but without the starter, it just doesn't go. It just doesn't start up. So we don't know. We don't know what it is we just lost. And because we don't know what it is we just lost when we lost life, we don't know how to bring it back again. So it's a very reasonable question without an answer.
Okay, and he's got another question there. It's in a couple parts. The phenomenon of orphan genes seems to be a compelling case for the design of life almost to the level of individual species. What is the current secular explanation for the origin of so many proteins? And if the answer is de novo proteins from a non-coding DNA, doesn't that beg the question of where these DNA sequences came from to create useful proteins? I assume they are rare.
Dr. Sanford and I, he had me tasked to this for like 1 or 2 years. And that does relate to the nylon eating bacteria problem. And then that's how I came up with the protein orchard argument. So the protein orchard argument is a stronger example of orphan genes. And the evolutionary biologists will use a lot of equivocation. So if you have a 2-character password, it's like, well, random chance can make that. But making, solving a password that's like 1,500 characters long, that's difficult. But they tend to equate the two. So you could get all these random transcripts. They'll call it a protein. They don't even have confirmation, because Dr. Sanford had me look at some of these claims of the non-coding regions. I wrote one of the researchers. We couldn't even determine if these things were even coded or if they are actually junk RNA sequences or what we call link RNA. There's so much we just don't know. But I would say you can't use the fact that a simple protein came up to explain far more complex proteins, because they all have, and I've talked to protein biologists about this, every protein has its own statistics. So people keep quoting the Doug Axe statistics on beta lactamase. That, there are proteins way more complex than a beta. It's very easy to make a beta lactamase. It's very hard to make a topoisomerase. So the numbers are even more astronomical for that. What I would do is stop, this whole thing about making these sweeping generalizations, trying to account for every protein on the planet. You're going to get burned on it. The better strategy to argue about this from an ID perspective is take select well-characterized proteins that are very, very well studied. And then you could say, hey, look, something this complex, why don't you give me an explanation? You can't just take some random non-coding DNA and say that it had a 50 amino acid sequence. We don't even know if it's just a junk sequence and whether it's usable. You can't use that to try to explain the emergence of something as complex as a topoisomerase. That's the strategy I would use. So what are their explanations? Some of them have said, we still don't know. I've seen the papers and it's problematic. I think Paul Nelson at the Discovery Institute would be the expert on orphan genes. That's his baby. He thinks that it's going to be very difficult. I do know that for major groups of creatures, collagen is like orphan gene. Without collagen, you wouldn't have bones and skin. And that's a complex protein. I showed you pictures of the sequence. I've challenged evolutionary biologists. I said, explain that. One thing I've noticed, they've rarely ever said appeal to non-coding sequences to explain that. I would just focus on well-characterized proteins. And there are a few of them that have had a lot of study by hundreds of researchers. Just focus on that. I don't like these questions that are like so sweeping, or these ideas or claims by either side. You're going to get burned. Stick to data that's well-characterized rather than trying to make universal claims. It's just too difficult. Just stick to small ones and just see how the other side reacts. That's what I'd suggest.
Awesome. And the last question here is, there was an example given that 6% of human proteins are novel relative to other primates. How could this ever be possible with only random mutation?
I want to tell you, we've only characterized only a fraction of the proteins out there. It's very difficult to sequence. It's easy to sequence the DNA and then do what we call protein predictions and say, maybe this is a protein, or really sequence the RNAs too. We still don't know a lot of what's out there. And there's some proteins expressed only in certain cell types. They're anywhere from hundreds to thousands or tens of thousands of cell types, depending on how you categorize it. We've certainly not tested all the possible cells of all the 37 million cells in an adult, much less all the cells in all the developmental phases. There is so much we don't know, and both sides are eager to make generalizations, and I think that's risky. I have been searching for truly unique proteins to humans and trying to see how well characterized they are. They're not very well characterized. It's way too early in the game I wouldn't feel comfortable to be making arguments about humans. I am comfortable talking about stuff for major groups of creatures. Creationists focus on chimps and human ancestry. That's a difficult transition, but you'll have an easier time. The easiest transitions to argue if you're a creationist or ID proponent is from prokaryote to eukaryote. You have very distinct proteins. They're generally very well characterized that distinguish them. So people are surprised I don't argue over human evolution. I say it's just too hard. Argue over prokaryote, eukaryote. It's just you're going to have an easy time. I will tell you this. Change Tan, she was a professor of molecular biology at Missouri. She came from communist China. She was an atheist. I think she definitely was not a Christian. She was asked by Nobel Prize winner George Smith to teach molecular biology. And in the process of studying the transition between prokaryotes and eukaryotes, she became a creationist. And that eventually cost her job. But all this to say, some of the people where I've seen dramatic conversions, I don't know anyone that has converted to being a creationist because they studied human evolution. I know several that became Christians because they studied the origin of life. And the next after that would be the prokaryote eukaryote transition. I would really focus on that.
Thank you so much, Sal. Before Kurt closes us off, I just wanted to say thank you guys for your time, for your patience and your humility and love for the Lord. Really appreciate all that you guys do, and I pray that the Lord's hand will be upon you in all your future endeavors, and that you continue to glorify Jesus.